Welcome everybody to the to part 3 of the 5th annual Montreal Oscillometry summer seminar. We've got a great program lined up, and rather than outline what's going to happen, I think we'll just get to it to maximize the discussion time. We will be a little bit less pressured, fortunately, this week than last week with 5, not 6 presentations. So again, the program is as you've got on the PDFs I've sent you. What's fun is some additional statistics which seem too good to be true, so I'm not quite sure, but when I review the traffic for the 2 weeks on my Zoom, and the only meetings I've been having have been the part 1 and 2, we have 321 participants cumulative, so that's an average of 160 each session. I suspect that's high because when I look at the gallery view, I see somewhere between 75 and 100 individuals. So maybe these are people coming and going in between, but I'll try and sort that out for next time. But certainly I think the 21 countries is an accurate representation of the coverage that we're having on a global basis. So thank you all for participating, and I have to get used to saying not only good morning, but good afternoon and good evening as we start the seminar. Also to remind you that the Respiratory Effectiveness Group, which over in the UK, has posted last week's seminar. So part 1 and 2 are available, and we'll try and continue posting on a weekly basis the remaining 2. And finally, a tip of the hat to our industry partners who make this seminar and the other work that we do at the Oscillometry unit here in Montreal possible. So with that, I'm going to turn the screen over to Shakira Shaya, who's from the University of Cape Town. And Shakira works with Diane's Gray's group, and she's generated enough data that we're going to give her room for 2 presentations. So we'll start with indoor pollution or tobacco smoke exposure and lung function at 3 years in an African birth cohort. And the discussant for that will be Julian Allen from the University of Pennsylvania down at CHOP. And for Shakira's second talk, the oscillometry in healthy South African children and adolescents, we'll have my good friend Larry Lands, who's from the Children's Hospital here in Montreal at McGill University, lead off the discussion for that. So Shakira, I see you've got the screen up and running. Well done. Welcome. And it's with great interest that I'll be listening to your talk, as will the rest of us. So thank you.
Hi, thank you for the introduction, and thank you for giving me the opportunity to present our work today. My slides don't seem to be moving.
Well, take your time. We're not behind yet, so it's okay.
I'm not sure why. I should maybe I am sharing my screen.
When you are indeed.
On screen view. There we go. I have no conflicts of interest. And some background. Childhood respiratory illness remains a major challenge for global health. Tobacco use is a leading cause of preventable disease globally. And as we know that antenatal exposure to maternal smoking, as well as indoor air pollution, impairs lung function. And postnatal exposure to both indoor air pollution and smoking in early childhood has been associated with respiratory disease and reduced lung function in later childhood. This is the Drakenstein Child Health Study. It's a birth cohort study which is led by Professor Heather Czar. This is situated in the Western Cape, and it's about 60 kilometers from Cape Town. This is a peri-urban area. It's a very low socioeconomic population, and there is a high infection disease burden, including HIV, as well as high indoor air pollution. There's a good primary healthcare system with a high vaccine coverage. Enrollment took place at 2 sites, which included the eMbakweni and TC Newman. And overall, this is a stable population with minimal movement. What do we already know from the Drakenstein Child Health Study? So from the studies that were done by Professors Gray and Anissa Venke, they have found that infants whose mothers smoked during pregnancy had lower lung functions compared to mothers who did not smoke during pregnancy. And having a mother who smoked during pregnancy actually trebled an infant's risk for having lung functions in the lowest quartile at 1 year. And antenatal exposures were much more strongly associated with respiratory disease in the first year of life. And antenatal maternal smoking, environmental tobacco smoke exposure, as well as particulate matter 10 exposure was associated with lower respiratory tract infections, wheezings, and hospitalizations for respiratory infections in the first year of life. The aim of this study was to assess the impact of postnatal indoor air pollution and environmental tobacco smoke on lung function and bronchodilator response at 3 years of age in an African birth cohort. Infants were enrolled, they had lung functions at 6 weeks and 3 years using the forced oscillation technique, and both standard measures and intra-breath measurements were done. In infants, the patients were supine and they had a face mask covering during the test, and patients were quietly asleep. 3 years, patients were sitting comfortably, their cheeks were supported, and their nose were clipped. Measurements were collected using the FOT equipment, which is the wave tube, and signals of 32 hertz for the standard measures and 10 hertz for the intra-breath measures were collected. And there was a minimum, we chose a minimum of 3 reproducible spectral measures and 1 16-second epoch of 10 hertz frequency. In the 3-year-old children, bronchodilator response was measured using 400 micrograms of salbutamol, which was given via a space mask and a spacer, and testing was repeated after 15 minutes. For the analysis, we only included intra-breath measures because they were the most useful. Looking at the indoor air pollutants, particulate matter 10, more than 40 micrograms per meter squared was considered high, and this is according to the WHO acceptable standard. Looking at environmental tobacco smoke exposure, this was assessed using infant urine cotinine, and the highest level was measured from 6 weeks to 3 years. No exposure is if the levels were less than 10 nanograms per mole, and moderate and high exposure if it was more than between 11 to 500 nanograms or more than 500 nanograms per mole, respectively. These measures, the measures for PM10 were collected antenatally and postnatally, and antenatally was between 28 to 32 weeks and postnatally between 4 and 6 months. The statistical analysis, we looked at linear regressions for the impact of the indoor air pollution, looking at particulate matter 10 and environmental tobacco smoke on the intra-breath measures at 3 years, and we explored both an unadjusted and adjusted model. The unadjusted model the adjusted model was adjusted for 6-week lung function, sex, socioeconomic status, the BMI Z-score for age, as well as site and previous pneumonia. There was a total of 768 children tested. We excluded 322 children for the following reasons and included a total of 446 3-year-old children. In terms of demographics, males and females were fairly equally distributed, with 47% being males. Even the sites were fairly equally distributed, with 51% and 49% between Mbekweni and TC Newman. About 6% of the population was underweight, and 19% were actually stunted. Children born between 32 weeks and 37 weeks were there were about 12% of children, and those less than 32 weeks were excluded. Children who were HIV exposed, these include 84 children, which were 19%, and no HIV positive children were included in the analysis. In terms of postnatal environmental PM10 exposures, 16% of children were above the ambient standard. About half of the children had at least one previous lower respiratory tract infection at the age at 3 years. And looking at tobacco smoke exposure, 17% of children had no exposure, and about 76% of children cumulatively between moderate and high exposure had some tobacco smoke exposure. Here we look at the resistance and reactance at 6 weeks and 3 years of age. At 6 weeks, we look at resistance and reactance as well as some of the intra-breath measures. And as we can see with 3 years, there is a decrease in resistance and an increase in reactance as we would expect. And within the 3-year age group, between the pre-bronchodilator and post-bronchodilator, we also notice a difference. And this is expected even in normal children. We look now at the impact of PM10 on pre-bronchodilator at 3 years. And we see that there is a significant increase in delta R in the unadjusted model, but not in the adjusted model. Delta R is the difference between resistance and inspiration minus the end expiration. And post-bronchodilator, looking at the PM10, we note that the delta R in the unadjusted model persists, the significant persists. There is also an increase in the adjusted model of resistance and expiration, and a decrease in reactance at end expiration, reactance at end inspiration, and X mean. Environmental tobacco smoke exposure pre-bronchodilator. In the unadjusted model, we noted there is an increase in resistance at end expiration. In post-bronchodilator, this persisted again only in the unadjusted model. Looking at the unadjusted model, we also note there was an increase in resistance at end inspiration and R mean, with a decrease in reactance at end inspiration and X mean. And on the adjusted model, the decrease in reactance at end inspiration persisted. So in summary, children exposed to high levels of PM10 had lower post-bronchodilator lung function compared to unexposed children. In the pre-bronchodilator lung function on the unadjusted model, we noted an increase in delta R and no effect on the adjusted model. And post-bronchodilator, on the unadjusted model, there was an increase in delta R, and on the adjusted model, we noted an increase in resistance at end expiration, a decrease in reactance at end expiration, at end inspiration, and also the mean reactance. Children exposed to high levels of postnatal environmental tobacco smoke exposure on the pre-bronchodilator lung function on the unadjusted model, we noted an increase in resistance at end expiration. In post-bronchodilator, on the unadjusted model, an increase in resistance at end expiration, at end inspiration, and the mean resistance, with a decrease in reactance at end inspiration and the mean reactance. And on the adjusted model, there was only a decrease in reactance at end inspiration that persisted. In conclusion, high exposure to environmental tobacco smoke or indoor air pollution in early life is associated with impaired lung function at 3 years, and this was adjusted for the 6-week lung function. And the effect of postnatal exposure was most notable after bronchodilator administration, which then suggests lung structural damage, which is a mechanism resulting in fixed airway impairment. And I'd like to acknowledge the following trusts and the individuals at Paul Hospital, as well as the families and children who participated in the study. Thank you very much.
Shakira, thank you very much for a lovely presentation. If you stop sharing your screen, you can go back to gallery view, and I'll hand the podium over to Julie and Alan. And I'll just remind everybody on the call, please mute your microphones. We had some background motor vehicle traffic and the occasional cough coming through during the talk. Okay, Julian, over to you.
Well, Shakira and Diane, that was a very polished and informative presentation. I really like it, and learned even more during our conversation yesterday. So I'm going to take some questions in a bit, but I just wanted to maybe frame some of these questions with a couple of observations. First of all, your results seem very robust. I think that physiologically, everything makes sense in terms of the changes that you see on resistance and reactance with growth, with inspiration, with expiration, and pre and post bronchodilator. So I think that lends a lot of credibility to your results. A few comments. In terms of the longitudinal nature, looking at results at 6 weeks and results at 3 years, of course, there's a difference in methodology in terms of a face mask used at the 3-week gauge and mouthpiece and nose clips at the 3-year age. And since infants are obligate nasal breathers, that may play into those results. But clearly, the difference between 6 weeks and 3 years is very large and not enough. And that difference between face mask and mouthpiece isn't enough to explain that difference. We talked a little bit about the indoor air pollution, and yesterday, this didn't come out in your presentation, but yesterday during our conversation, I thought it was very interesting that that often in some of the lower socioeconomic strata, that when the electricity runs out, there's a conversion from electric heat to kerosene heat. So it'd be interesting to look at studies that were done at the beginning of the month, studies that were done at the end of the month, to see if there's an effect of that in terms of the ambient air pollution. The other comment I wanted to make had to do with your observation about the stunting in these groups. So it wasn't trivial. I mean, on average, there was a standard deviation difference between height in these infants, and I think that between infants and normal infants, and your model adjusts for the lung function at 6 weeks, socioeconomic status, and body mass index. And one thing I would suggest as a very interesting thing to look at in the future is also just taking a look at the effect of height, not body mass index, but the effect of height, since at the age of 3 years, arguably, height is a more robust predictor of lung function than body mass index is. And the last thing I wanted to address, which I think is sort of the meat of your presentation, are these use of the adjusted versus the unadjusted models. I think this was really brilliant, and I think it actually, some interesting lessons fall out from that. I think the adjusted model suggests that lung function at the age of 6 weeks is perhaps a more important predictor than either environmental air pollution or exposure to or indoor air pollution or exposure to smoke. And the adjusted models suggest that PM10 is the main driver of lung function difference, whereas the unadjusted models suggest that factors other than environmental tobacco smoke are the main driver of lung function difference. So this is my interpretation. I'm not a statistician, but I wonder if you or anybody in the audience has any comments of that PM10 perhaps seems like a more important driver than environmental tobacco smoke. So with that, I'll stop and see if anybody else has any questions or comments. And Shakira, clearly I'm interested if you have any responses to the things that I've brought up and any other insights that I've not mentioned.
Thank you very much. And thanks for your valuable insight. And I think going back and looking at height alone will be very interesting to see. And I think to mention that these models are actually quite complex, or actually the human model is quite complex. So I think there's many other many other factors that can contribute to these changes. And we try to account for what would be the most common, but I think it just highlights how complex this is. And we know that there is associations, but many of the confounders are we still need to address, or they are something that we still don't know what it is that's actually accounting for these changes.
Well, I really like your comparisons of adjusted and unadjusted models. I think there's a lot of insights to be gained from those.
Yeah.
Any other questions from anybody else in the audience?
So I'd like to echo Jules's comments. I thought that was a great presentation and very interesting. Yeah, a couple of things. One, I was curious, what was the nature of the indoor pollution? And now I'm hearing it's probably kerosene and not necessarily biomass.
So in terms of, we looked particularly at PM10, but looking at what patients do have, about 90% of the population do have access to electricity. But as Julian mentioned, that towards the end of the month, they often go to alternate fuel burning, and that includes wood, fire, paraffin, and very few coal. And generally, it's was more noted within the eMbokwene population rather than the TC Newman. Yeah, so about 30% of those sometimes would burn alternate fuels.
And the other thing I was wondering, and it goes along with towards the adjusted and unadjusted model. I was curious how well the 6-week values track. In other words, if you start off small, generally most data suggested you stay small, unless you have an exposure, and then you could get even smaller. And if you start off normal, unless there's major exposure, you should track normally. And I'm just wondering if there's a way of looking at that tracking from an early age.
Yes, that's the plan. And we are busy looking at those models, and we're assessing the tracking at different time points. It hasn't presented here, but from what we think and what we know at this point, it'll probably still be within the lowest quartile, although we still need to show this formally and statistically, yes.
Hi, it's Di here. Julian, are you happy for me just to make a comment quickly?
Absolutely.
Yeah, thanks, Shakira, for presenting that so succinctly. Really nice presentation. Just to comment on the tracking of the lung function from, so we certainly have shown very clear tracking from 6 weeks up until this time period already. So the children that are in the lowest quartile stay in the lowest quartile, very similar to what's been shown in other birth cohorts. And it was certainly one of the reasons why we thought important to adjust for that. But I agree with your insights, Julian, that perhaps the mechanisms are slightly different between these air pollutants, and that tobacco smoke antenatally seems to have a very strong effect. So lung function being abnormal really soon after birth. And that perhaps the major impact has happened by that point, and the environmental, potentially, maybe the environmental impact is less than the antenatal impact. It's not, you can't say that for sure from this data, but suggestive. Whereas the PM10 certainly is a concern. I think this really just flags the importance of air pollution in normal and healthy lung growth and development and risk. We've also shown that the lower respiratory tract infection impacts your lung function independent to many of these other factors. And Nisa Vanka has shown that PM10 exposure increases your risk to lower respiratory tract infection. So that really speaks to how these things interact to cause damage. But I suppose the bottom line is it's not good for healthy lung growth and development. And it could really be used in advocacy and trying to get our most vulnerable children into safer environments.
Yeah, thanks for that, Diane. And I'm not a statistician, so this question may be naive, but when I looked at these data, I sort of came away with the impression that PM10 was more important than environmental tobacco smoke. And I realize it's not a competition, but is that a misinterpretation of the data to think that postnatal tobacco smoke is less of a driver than the PM10 in terms of these lung function differences?
So Julie, I think it's a difficult one to answer from this data, because one thing about this cohort is there's a huge amount of tobacco smoke exposure. I don't know if you noticed that. So, actually over 70% of the children have some environmental tobacco smoke, and they live in small homes. So there's quite a lot of exposure. And we saw a dose effect. So the more exposed, the more significant the impact, as well as tobacco smoke being one of the contributors towards the high PM10. So I'm not 100% sure. I think that I interpreted that the antenatal impact of mum smoking is sort of maybe a stronger effect on the baseline lung function, and we've corrected for that. Yeah.
Thank you.
That's a question I wanted to ask, is to what extent is the PM10 and environmental tobacco smoke overlapping? I mean, how much of the PM10 consists of environmental tobacco smoke? I would think not an insignificant part given this description of the home environment.
Mm. Yeah, thanks, Charles. I mean, that's a great question. Shakira, I don't know if you want me to answer, or would you like to have a... We did look, you know, we have looked at the correlation between PM10 and tobacco smoke exposure, and it doesn't actually correlate as well as you think. So I certainly think there are other contributors other than the tobacco smoke, but
Okay.
it's very difficult to
Well, I
tease it
think
out
that
in time.
the cotinine level should give you some insight to that, which is what I'm presuming you're talking about.
Yeah, that's right. So the cotinine didn't correlate as strongly, you know, completely. I can't remember. Sorry, I don't have the data in the foreground. I'm not sure if Anissa Bank is on this call. She might want to give more insight, but it's it wasn't just the tobacco smoke, the PM10.
Yeah, no,
yeah.
I get that, but I was just curious to what extent, and how do you actually do that in the homes? I'm just curious on a technical level. Do you put some sort of sensor in the room, or how does that work?
Hi, Di. I am on the call. Should I jump in and help?
No, go for it, Anissa.
It's
That'd be
Anissa,
lovely.
thank you.
Yeah.
Yeah, please
Hi,
turn on your video, folks, when you're talking. That goes Charlie and Anita, please.
Yeah, well, it's not a
Sure,
pretty
I'm
picture
trying.
this morning.
Don't.
Yeah.
There you go. Hi, Charlie.
I'm struggling a bit. Has it come on or not?
No. Don't worry about it.
But go ahead, please.
I'm having
Let
a
me just
bad
go,
hair
sorry.
day, so don't worry about it.
I'll put it on in a second. So the way it was measured was through using, at that stage, we'd use actually personal air check sensors left in the homes for 24 hours.
Okay.
And what we had was pre-weighed filters and then left 48 hours and then collected after that time and then sent to lab to post-weigh and analyze. So yeah, that was done both in the antenatal and postnatal period. To add to that, yes, although it wasn't correlated, but yes, there is the thought that some of the smoke exposure does contribute to particulate matter, but there are other factors as well. So I think it's not just the tobacco smoke that's contributing. I'm sorry, I'll try and sort camera out in a second.
Could I add a comment? This is Jeff Fredberg. I was impressed that you can detect these differences using oscillometry. And aside from cigarette smoke and kerosene burning, in Massachusetts now, there's a big push to outlaw natural gas cooking because it releases nitric oxides into the room. And I know a person in my department who's studying it, they find health effects and asthma triggers from natural gas. So there's a big push to outlaw natural gas. I think that it'd be interesting to look at what that person's done and whether your techniques would be useful in a kind of a different context. So thanks for that.
And in Vermont, there's a big push to get rid of bad wood-fired or bad pellet stoves that off-gas all kinds of crazy things. So these are both, you know, things in the home environment that could be pretty nasty. In kitties, you know, that are growing and then being exposed to so the interaction between cigarette smoke and PM10 could also be of interest. And you have the data to look at that.
Yeah, thanks.
Jules, could I jump in with one more question before we move on so Shakira could get her second talk in? Clearly, it's of interest with the discussion you're generating, Shakira. Congratulations. The question you might punt to Zoli or Diane if you can't answer it. I've been messing around in my own practice with Zoli's intra-breath technique, and we've been looking at loops of the reactance versus volume and reactance versus flow. You do have that data. Have you looked at it yet? And does that give you any added value to the findings that you presented?
I personally haven't looked at it, but I think I'm going to jump to Di or Zali and see if they have.
we haven't looked at it in detail yet, but I would love to give Zolly an opportunity to comment, because I mean, just to say that, I mean, it is wonderful that we are able to detect these relatively small changes with FOT, because we did comprehensive lung function measures in these infants, and oscillometry was certainly seemed to be the most sensitive to picking up these changes. And we haven't even done it justice yet. So I'm not sure, Zolly, if you would like to also have a word.
Yes, the reactance changes during inspiration and expiration, which normally form a very slight hysteresis, not terribly exciting. This is exaggerated in the presence of expiratory flow limitation. So we see these in obese patients, otherwise normal. We, of course, see this COPD, asthma, and other pathologies. In 6-week-old infants, we occasionally find this looping. But in the present population, at 3 years old, it is not very common. It doesn't seem to indicate any expiratory flow limitation. It is more probably having to do with the inspiratory change of reactance, which is the delta X, which might reflect some change in the inhomogeneity during tidal breathing, but it is not very apparent in this population.
Okay, thanks. Well then, Shakira, why don't you share your screen again and give us your talk titled Oscillometry in Healthy South African Children and Adolescents. And then Larry, you can take over after that with the discussion.
Okay, thank you for the second presentation. So the background, measurements of lung function is important for the diagnosis and management of lung disease. And as we know that spirometry is the most commonly performed lung function, but we are all aware of the limitations in younger children because of the technical difficulty and also the relative insensitivity to early lung function. And oscillometry is feasible for all age groups, including young children. And we can measure both standard measurements, which include resistance and reactance, as well as we can do a bronchodilator response. And then we also have intra-breath measures, which detect changes in the respiratory resistance and reactance that occur within the breathing cycle. And it's more sensitive with respect to airway obstruction, as well as ventilation inhomogeneity. And one can actually pick up respiratory disease risks more than the standard measurements. And the clinical application is limited in Africa because of the lack of reference data for non-Caucasian children. And this is despite the high respiratory disease burden. The majority of current oscillometry reference data are specific for Caucasian populations. And this age range is between 2 and 16 years of age. Non-Caucasian studies include a Mexican cohort, an Indian cohort, Thai, as well as an Emirati populations. And here, the age range ranges between 3 to 12 years. In these studies, the data was collected with a different commercial equipment, and it was also poorly standardized. And as I mentioned before, there's no reference data in African populations. The aim of the study was to develop reference range equations for oscillometry in South African children between the ages of 3 to 17 years of age. The participants included healthy children from the Drakenstein birth cohort, as I mentioned the cohort before. The additional patients came from the Cape Town adolescent antiretroviral cohort. These were the healthy controls, as well as healthy patients attending the surgical outpatient clinic. The equipment that we used was the InCircle FOT wave tube system, and we collected both standard measures and intra-breath measurements. And children were seated comfortably, as we can see, with the nose clipped, and the cheeks firmly supported, and they took a breath through a mouthpiece as well as a filter. Patients with more than one testing point were randomized to include data from only one time point per individual. From the statistics point of view, the relationship between the oscillometry variables and sex, age, weight, height, and ancestry were explored using backward stepwise linear regressions, and a reference range equation for each variable was generated. The effect of collinearity was addressed, and the equation we used height only, as the addition of other variables showed negligible changes in the adjusted R squared. The effect of puberty was also addressed, and this is where we generated equations for the Drakenstein child health children, and this was children between the ages of 3 to 8 years of age, and once comparing to the reference population between the ages of 3 to 17 years, the adjusted R squared was also negligible. Demographically, totally we included 710 children, and approximately 48% were females. A majority of the patients, as we can see, came from the Drakenstein child health study, and between the ancestry, they were fairly equally distributed. And below also, we looked at the anthropometry measures for the population. This is a summary of the respiratory variables. Here we included the spectral measures and the intra-breath measures. These included the resistance and reactance, as well as the intra-breath measures of resistance at an inspiration and expiration, as well as reactance at an inspiration and expiration. Plotting the cohort of patients of using resistance against height. As we can see, with increased height, which is also associated with an increased age, we note there's a decrease in resistance. And looking at reactance versus height, this is the second graph. This is more variable, but we do note an increase in reactance with an increase in height. The reference range for standard oscillometry measures in our population, here we included resistance and reactance, as well as resistance between R6 and R20, and area under the reactance curve. And looking at the intra-breath oscillometry variables for our population, here we looked at resistance and reactance at an inspiration and expiration, as well as the R mean and X mean. And looking at delta R, which is the resistance between which is the variables between resistance and inspiration minus expiration, and similarly with the delta X. Looking at the comparison between our current equation and other reference equations, looking specifically at 6 Hz, the dark blue line, if you can see my arrow, represents our equation. And as we can see, at heights below 120 centimeters, our patients have a higher resistance at 6 Hz compared to the other populations. Below our population, on the red and the orange line, we see the Shackleton reference range equation and the Albushi equations. And the Shackleton equation looked at a Mexican population and the Albushi population and an Emirati population. Below that, all the reference equations were equations developed for Caucasian children. Well, the light blue broken line is the population from the study Udapata-Pong, and they looked particularly at, this was a Thai population, and this was completely out. So looking at the strengths of our study, this was a large sample size. Each child appeared at only one time point. Patients were tested using the same equipment. The testing was performed by a consistent trained team of 3 people, and methods and testing procedures were standardized across all the sites. The limitations, there were low patient numbers in the 8 to 16-year age group. Black African children were overrepresented in the 14 to 17-year age group. And this reference equation may only includes data from the Western Cape, and it may not be generalizable to the rest of the southern region. And this equation needs to be tested in different population groups and settings. In conclusion, oscillometry outcomes in healthy African children were inconsistent with existing reference data. And this highlights the need for population-specific reference data in the absence of appropriate, robust international standard. And we are currently all working towards a universal, internationally defined equation. And this will facilitate early identification and management of respiratory disease. So what is the way forward? Currently, there are many commercial equipment available, as we can see and as we're all aware of. Many include impulse oscillometry and also the normal sinusoidal patterns. And currently, there is the harmonization group, and this group has been formed to allow the standardization of equipment and to find agreement between commercial equipment and the wave tube. And the ultimate aim, as I mentioned, is to develop a universal reference equation across all commercial equipment. And as we currently know, there is differences between the various commercial equipments. Thank you very much. And I'd like to acknowledge the following trusts and all the individuals involved in the collection of the data, as well as helping us with the analysis. And most importantly, the staff in the Drakenstein area and the patients. Thank you.
Well, Shakira, thank you for a second fabulous presentation of a strong project. Larry, I'll turn the podium over to you to lead out the discussion.
Yeah, Shakira, again, once again, that was a great presentation. It was interesting data. I know you had fewer data at some of the older age points, but I was struck by the increase in variation at the younger ages compared to the older children. I really wasn't sure of what that was due to, whether it was just numbers or is there more variability in younger children. And I was, again, in reference to your previous talk, the influence potentially of stunting and or pollution that could be impacting some of those numbers. Certainly from Ron and Zoli's data and data that we've done comparing devices, I think certainly some of it is going to be equipment differences rather than, let's say, population differences or something, things like this. But it is interesting that, especially in your shorter or younger children, you had much more resistance. And I'm just wondering again, air pollution and stunting may be playing a role there.
Thank you, Larry. I absolutely agree. I think, you hit the nail on the head. Perhaps, we do know this is a low socioeconomic population, so stunting certainly can contribute to this. And the other point to mention is that we did have predominantly more younger children compared to older children. So this could account for the change. As we noted in that table, that the effect wasn't sustained. So if we're looking at things like exposures, like environmental tobacco exposure, one would based on the previous explanations, we would expect that this effect would be sustained. But as we noted in the older children, that this wasn't the case. So perhaps we have to look with more children and see if the lower children in the lower amount of children in the larger age group has in fact contributed to this.
Great. I'd like to open it up. I understand, Ken Berger, you recently have some normative data, and would you like to comment?
Yeah, I'll be happy to. Yeah, our data were in adults, so a little bit different. There's a couple of things I noticed in your data. One is you mention ancestry. And I'm curious as to the approach. This is something that a little bit of a pet interest of mine in terms of this issue about race and ethnicity, and which some might consider a social construct. And so I'm curious as to how that was dealt with in your data set, and you didn't show any information as to whether there were differences across groups. And our group was predominantly Caucasian, but we saw no real effect of race, self-identified race ethnicity on our normative data. And the other comment is your final models were height related and not weight related, but in the adults, we certainly know that weight has a huge impact on oscillometry data. And I don't know if that's unique to your cohort in South Africa. Maybe you just don't have obesity, I don't know. But I raised those two questions, I'm just curious.
Okay. With ethnicity, we noticed that we do have two specific population groups, and we wanted to assess if there was a difference, and that's why the ethnicity we put into the regression model to see if that would account for any of the changes. And maybe with one or two variables, it did make a difference, but overall, once comparing with and without, did not make a difference. And the second factor is that we did include height, but at one point included both, we included BMI, which includes weight and height. But in effect for the collinearity, we decided to choose one. However, saying that, when including the BMI with and without the height didn't make any difference to the model or the adjusted R-squared. It was like a negligible difference.
That was different in our data.
And looking in terms of obesity, I think, I think our problem would be more the underweight rather than the obesity or the overweight children. So we do have, and as we saw from the previous presentation, and overall that our children may indeed be underweight and as a result, chronically stunted. However, what's quite interesting is the effect didn't persist in the older age group. And we need to assess why and look at other factors that could contribute to this.
Yeah, I saw that you had corrected for ancestry, and I was actually taken aback that it didn't have an impact. There was some interesting work out of Puerto Rico where they tried to apply in terms of spirometry, looking at the global lung initiative values, and found it was all over the map, and they have a very mixed population. So they went to an ancestry-based predictive equations, and that actually were able to create sort of unique equations for Puerto Rico because their population just does not fit with, let's say, a US population, even though they're a protectorate of the US. So is it because you only had limited numbers in certain ethnic groups, or why do you think ancestry didn't play out in your equations?
When we look at, in terms of the ethnicity, they were equally distributed in our population. And looking at a few equations, I think maybe one or two variables, it did show some significance, but it, as I said, it didn't persist. So maybe the difference would be body habitus and height, which we did consider. So that could probably be the one thing that would be a commonality, and we accounted for that. But I'm not sure of any other reason why.
I think
could I add something? Could I just add something on the cohort, Charles, before you?
Yeah, sorry.
I just wanted to make the point that our cohort is all were all African, so they were all Black African, some of mixed ancestry and some less. But I mean, we didn't have any Caucasian or Asian in our cohort. So actually, from your point of view, this was really one ethnicity. And that is one of the reasons. But I just wanted to add that we have recently prospectively looked at spirometry across our population, because we have a very multi-ethnic population in South Africa, and did find differences between ethnic groups on the GLI reference equation. So, we are working towards international ethnic, non-ethnic reference equations, but we're going to need very big, robust data sets. And I mean, it does obviously speak to how other factors play a role. And if you can see that in this population, the kind of exposures, stunting, etc., plays a role that might have little to do with ethnicity, but more to do with socioeconomic factors. So I mean, these are complex issues, but that I think would be one of the reasons why ethnicity wasn't important here.
I just wonder if I could just add one comment. I'm not sure I agree with you totally about the social economic factors, and I wonder how much of the historic differences across race ethnicities are entirely related to a variety of other factors that are loosely captured by this self-identified race. And I think that's a huge topic, maybe beyond the scope of an oscillometry meeting, Ron.
Indeed.
I have some strong feelings in this area, and I think that largely because the confidence interval for most of the parameters we're predicting is very large compared to the small adjustments for race-specific equations, and that I think should be thought about, and underlying factors, which may not be, I don't think self-identified race ethnicity captures anything that's ancestral.
Well, Ken, I'll take it one step further and just urge everybody to get their hands on the book Breathing Race into the Machine.
Yeah.
A wonderful review of the history and the intimate relation with race and possibly abuse of spirometry to propagate certain ideas which hopefully are fading away. Really, it's worth the read. Charlie, over to you.
Well, there's a number of us on the call that are part of a working group addressing the issues of race and ethnicity and lung function. And it is an incredibly complex topic. In the United States in particular, it's a very sensitive topic at the moment, as everybody could probably easily appreciate. I think what's really exciting about this particular data set is that it suggests that oscillometry could be much more race ethnicity agnostic, that if I interpret your data correctly. And that's very exciting. And it suggests that if the differences that are seen with spirometry are really all, when you boil it down, because the FEV1, FVC ratios are not affected, to the size of the lung. And oscillometry, in a sense, gets away from that. And I think that's really very cool. The other question I had for Shakira, which, by the way, both these talks are terrific, is in your data set, I noticed that the resistance at smaller kitty size is higher. Do you think that's related to their environmental tobacco smoke and PM10? And if so, you could probably adjust it a little bit for that, and that would certainly bring it in closer to some of the other studies. Or did I get that wrong?
No, it was something that, thank you for the question. Yes, it was something that we did consider. And I think it would be a good idea to adjust for it. The only point against it is that this effect wasn't sustained. So in the older children, which we noted that there was a small number of children, but they resistance was not that high compared to the other populations. So perhaps it was based only on the younger children.
Yeah, but that would also be the reason for the higher resistance at smaller heights. So
yes.
Yeah.
However, the fact that it wasn't sustained, it's probably, we need to look into it and see if particularly how much of an effect PM10 and environmental.
Nice, but nicely done anyway. Thank you.
Thank you.
Larry, how about we take one more question from Zoli and then move on?
Absolutely.
Just a short reflection to Charlie's point. In fact, these normal values collected in different populations and with different equipment also differed in the fitting curve. So many normative data are approached with a linear dependence on height, which is nonsensical, of course. So that exaggerates the difference at low the height ranges. This is one. And the other point, just to be a little bit skeptical, maybe ruining the value of this very, very good talk and study a bit. I go back to Larry's first comment. I also have the concern that the differences between different populations are less ethnically rather than equipment-wise created. So standardization or harmonization is absolutely essential in the pediatric range where the impedance values are high and many equipment are not very accurate at high impedance measurements. I think not all to praise our study, but Jeff and Julian will agree with me on the accuracy of the wave tube measurement because they were pioneers in this technique. Thank you.
Okay, great. So thank you for giving us a strong start to this week's seminar, Shakira. Folks, you're in for a treat. Fasten your seatbelts because Ramon Ferre from Barcelona is going to take oscillometry to a whole new level as he explains cellular oscillation mechanics, followed by Jeff Fredberg, who will lead off the discussion. So gentlemen, the screen is over to you. Thank you very much for talking to us. Ramon, you're still on mute and you'll need to share your screen.
Can you see my screen?
Not yet. We still see you, but we can hear you too.
Now?
No, not yet.
Let's see. Now?
Yes, we now see your screen, but not yet presentation. Perfect.
Okay. Hi, good morning. Dear colleagues, in animal model research and in humans, oscillometry is easily measured from mouth pressure and flow. The quotient between pressure and flow is the mechanical impedance of the whole respiratory system. And this respiratory impedance depends on airways and tissue mechanics, and ultimately on cells, which are mechanically active and also secrete the extracellular matrix of tissues, pointing to the interest of measuring cell mechanics, for instance, by oscillometry. However, such a measurement requires to induce a controlled cell oscillation and to measure displacement and force at the scales of nanometer and piconewton. It took intense technological development and, more important, to change the biology paradigm to accept the role played by cell mechanics in pathophysiology. Approximately at the end of the 20th century, cellular oscillometry and more generally cellular mechanics experienced a considerable development. Today, I will present a short and non-comprehensive perspective addressed not to experts in cell mechanics, but just as an introduction to the topic. I thank the organizers for inviting me to give this talk, and I also particularly honored since the discussant of this presentation is Jeff Fredberg, a pioneer and a world leader in the topic, author of many seminal papers in the field. I will present and very briefly discuss three techniques to carry out cell oscillometry: magnetic twisted cytometry, optical tweezers, and atomic force microscopy. In magnetic twisted cytometry, cell is oscillated and assessed by magnetic tweezers. A ferromagnetic bit is attached to the membrane of the cell and is magnetized to be converted into a magnet, and then a perpendicular oscillating magnetic field is applied, who would tend to turn and twist the bit. The bit twisting is limited by the mechanical resistance of the attachment to the cell. So the stiffer the cell, the smaller the displacement of the bit. This bit can be measured by means of the image of the bit taken from above from a microscope and calculating the centroid of the image. And the torque applied to the bit is known because the magnitude of the magnetic field is known, and also the magnetic moment of the bit is known. So from the torque applied and the measured displacement, a stiffness index can be computed. It's a complex magnitude with a real part, G prime, and imaginary part, G double prime. It is interesting to consider what is the difference or the relationship between respiratory impedance measured by forced oscillation technique in patients, for instance, as we talked in the previous presentations, in which Z is computed as the quotient between pressure and flow, and then the impedance has a real part, which is resistance, and imaginary part, which takes account of the elastance. But in the case of comparing this magnitude G and Z, we have to take into account that in this case, there is a displacement magnitude, and here is a derivative, time derivative of a size magnitude, the volume. Taking into account the relationship between the Fourier transform of a magnitude and its derivative, it results that in the case of G, G prime represents the elastic component and G double prime the resistive component. The impedance or the stiffness measured by magnetic twisting cytometry is determined because the bit, you can see here, the bit here is attached to the cytoskeleton of the cell through the focal adhesions in the membrane. In this seminar paper by the group of Jeff Friedberg, this was seminal because they revealed the general behavior of the stiffness of the cells. You can see here the G prime and G double prime of cells subjected to different treatments, and you see that all the responses converge to a unique function, which is named structural damping, which is a kind of behavior already known in engineering. According to this structural damping theory, both G and eta hystericity, which is the quotient between G double prime and G, are related to X, which is a parameter that determines the slope of this frequency resistance of G prime. To illustrate some practical applications of this magnetic twisting cytometry, please let's see some examples about how histamine and thrombin modulate the G prime and double prime of the alveolar epithelial cells. You see here the control at what happens after addition of histamine and thrombin, which modifies considerably the stiffness of the cell. And also here in that example showing that pretreatment of the cells with activated C protein produces the thrombin-induced cell stiffening. You see here not pretreatment, this is the increase caused by thrombin. When the cells were pretreated with activated C protein, this increase was very, very decreased. This other example shows how this magnetic twisting cytometry technique can be combined with application of cell stretch. By joining the two experimental settings, you can see here a culture with cells and the bits before and after application of a stretch. And this experiment allowed us to see that the stretch induced stiffening in alveolar epithelial cells. You see for different levels of stretch, you see that both G prime and G double prime increased and that hystericity decreased. Interestingly, treating the cells with thrombin decreased the effect induced by stretch. You can see here without thrombin, this is the baseline, you see the increase in G prime, and after thrombin it was decreased, and the same for hystericity. To finalize with examples of magnetic twisting cytometry, this is a more recent example where you can see that mesenchymal stem cell cortical shear modulus scales with cell volume, which at the same time modulates cell differentiation. The experiment consisted on changing the cell volume by different procedures, by changing substrate stiffness, pattern sizing, or different osmotic tests, and to compute the cortical, the G prime of the cell for these different combinations. When all these data were put together, they collapsed in a unique relationship, showing that the shear stress module is related directly to cell volume independently of the method used to modify the cell volume. Now let's move to optical twisting cytometry. In this case, the oscillation of the bit is not produced by a magnetic twister, by optical tweezers. In that case, you can see here the cell, the bit attached to the surface of the cell. And in this case, this bit is not displaced by a magnetic field, but by a laser, by a light field. This is a laser beam that traps the bit, and then displacing of the laser may allow to manipulate the bit to displace it according to our frequency of interest. If one compares the displacement of the bit attached to the cell to a reference bit, which is on the ground, you can measure the stiffness of this cell. Interestingly, this technique can only be applied not only to bits attached to the membrane, but also it can be applied in the laser trapping intracellular organelles, so that is a more direct way to measure the cytoskeletal or the intracellular stiffness. Here you have a very clear illustration of the method. You can see here the amplitude oscillation of the reference bit at different frequencies of oscillation, and here the corresponding amplitude oscillation of the bit attached to the cell, also at different frequencies. Here the loops corresponding to the cell and reference displacement. And from this data, it's possible to compute, again, the G, the elastic modulus, the complex elastic modulus for different, in that case, the author applied the technique to different cells, fibroblasts, astrocytes, and neurons, and in all cases, they recovered the known structural damping behavior of the cell. Now briefly move to atomic force microscopy. In that case, cell oscillometry is applied by nanoindentation. So this method does not depend on a bit attached to the cell, but it measures directly the stiffness of the cells. In fact, the principle is the same that you use when we use our finger to touch an object to test whether it's stiff or soft. We apply a force that we control with our finger, and we observe the deformation of the object. In that case, the finger is a cantilever with a tip, which is pressing to the cell. In that way, we have that for a given force of indentation, there is a magnitude of indentation, and this can be measured a little bit complex with a laser beam and so on, but I will not go into detail. The fact is that force can be measured and the indentation can be derived by using a geometric model that relates the magnitude of force and the indentation. Of course, this relationship depends on the geometry of the tip. If in a maneuver of indentation, we fix indentation at a certain point and then oscillate slightly at a given frequency, a small amplitude, we oscillate the cantilever, we can record the signals of indentation and force at this given frequency to be able to compute what are the complex elastic modulus of the cell at this point. The complex shear modulus that is computed is the quotient between the frequency component of the frequencies transformed signal of force and indentation, and also using, of course, the geometry equation of the tip. Here, once again, the typical response, the typical frequency response of G prime and G double prime characteristic of cells, measured in that case in A549 cells, epithelial cells by IFM. Here you see an example of one of the advantages of IFM. As you put the tip of the cantilever at the place you desire with a microscope, you can select the exact place of the cell where you are measuring the stiffness. And for instance, you can measure at the nuclear site, at the nuclear area or at the periphery. And you can see here clearly that there is a difference in the stiffness depending on the place, on the site of the cell you are measuring. In particular, in this experiment, they observed that this effect of the site depends on the temperature of the cell. Final example of atomic force microscopy application was to, for instance, to detect that neutrophil activation induced cell stiffening. This illustrates the fact that the tip of the cantilever in AFM can be, for instance, spherical and can be applied to a spherical sample. In that case, is a non-adherent neutrophile, and using the adequate geometric model for indentation, the technique can be applied, and in that case, produces the result that the activated neutrophiles, you can see here, that are really more stiffer than the passive neutrophiles. The point now is all these techniques, how can they be compared? Well, the first answer is that all techniques provide different and useful information because are based on slightly different principles. And there are some specific differences. In the case of magnetic twisting cytometry, many cells placed at many cell sites are measured in a single maneuver. This provides good statistics because hundreds of bits are measured at the same time, but poor definition because the bits are distributed at random at different sites of the cell. A characteristic also of MTC is that the G obtained, the elastic complex modulus obtained, depends on the degree of bit attachment to the cytoskeleton through focal adhesions, and also that the fact that the cortical cytoskeleton is remodeled by bit addition. So to a certain extent, the technique modifies the previous state of the cell. AFM, on the contrary, carries out one single cell in one single site per measurement. So it's more detailed. This results in poor statistics. You require much more time to carry it out an exploration, but you have very good definition around, about the place of the cell you are measuring. Of course, the value of G depends on the adequacy of the indentation model. But I think that one of the important characteristics of AFM is that it allows to obtain a very detailed map of stiffness and fluidity derived from G prime and double prime, as shown in the case of this fibroblast in a relatively recent publication. So I think that this is a characteristic of AFM to obtain detailed maps, geographical maps of the stiffness and fluidity of the cell. And before finishing, I would like to mention some practical difficulties which are important in the real world about reproducibility, standardization, and device availability. Because usually these techniques, for instance, MTC and AFM, are carried out with very complex home devices, which are available only for a few number of labs in the world. And each lab uses their own criteria for computation and data processing, with no standardization and with difficulty, therefore, to reproduce and to compare the different results. Fortunately, at least in the case of AFM, there are initiatives trying to standardize the measurement, something similar to what happened or have happened in the past with the forced oscillation for measuring lung impedance. For instance, in this recent initiative, 8 expert labs working on measurement of AFM on cells, they measured the same gel of reference, so a reference gel. And you see that each of the 8 labs obtained different values with a certain variability. That depended or on the device they used or on the criteria they used for data processing. After taking a given protocol, which was agreed among the experts, you can see that this big variability that was observed from, let's say, spontaneous data processing, this variability was reduced. So I think that this is a good step to standardize the conditions and to be able to advance in the comparison of the results of the technique. And there is also the problem of availability. So as I mentioned, few labs in the world are able to dispose of these devices for research. In particular, in the case of AFM, which is also known as scan force microscopy of nano indentation, these are different names that with different small differences, but at the end, they are the same principle, so are equivalent. So I think that is interesting to see that now, very recently, there are industries providing to the market affordable, automated, and high throughput measurements for nano indentation of AFM, as you like. Here, this is an example. You can see a compact device. I have deleted the logos because I'm not recommending nor promoting this device. I'm just saying that this is an initiative in the good direction. Is a compact device when you can see here, then it can, in a conventional well of cultured cell, the device is moving automatically the cantilever, the sensor, and it is moving in a continuous way You can spend overnight measurements because all the setting is under temperature and oxygen and CO2 concentration, control it. So I think it's a good initiative because this measurement is free of operator variability. There are not arbitrary criteria. And I think that is a good direction for facilitating the expansion of cell oscillometry to different labs. In that case, for instance, the device allows to superimpose different frequencies at a given indentation and modulate the frequency and amplitude of the different oscillations. And this is the force measured by the tip corresponding to different frequencies. Okay, so this brief presentation just to conclude the following. Cell mechanics, and in particular oscillometry, can be measured by several techniques, each providing different and complementary information. These techniques can be used to investigate both fundamental cell properties and also practical questions on the response of cell mechanics to different pathophysiological stimuli. It is expected that new device developments may make easier to carry out automated and high throughput measurements for translational and even clinical applications. Well, in an editorial, recent editorial written by Peter Calverley and myself concerning the application of the oscillometry in patients, the conventional technique, we finished the editorial writing. Hopefully, more use in patients will trigger the interest of industry to produce more simple and affordable devices in a virtuous cycle leading to extended application of oscillometry in the clinical arena. I would be happy if in a few years someone could write something like, hopefully more use in cells will trigger the interest of industry to produce more simple and affordable devices in a virtuous circle leading to extended application of cell oscillometry in the translational research arena. Just to finish, to acknowledge my colleague and friend Daniel Navajas, who has been the leader of this line of research in our lab for the last 2 decades. Thank you very much.
Ramon, thank you very, very much for a stellar presentation. And Jeff, we are looking forward to your comments and leading off the discussion. So over to you in Boston.
Okay. Ramon, thank you for that exciting presentation. All of this came out of oscillometry, the way we currently understand it. It was back in the early, I just want to add a little historical context. It was back in the early 1990s. Jeff Maxim came to my lab, Ben Fabry, and we started wondering what is, this tissue resistance that we all know that decreases hyperbolically but frequently. What is going on with that? And we wanted to understand mechanism. And we said, well, we wonder if it's a cellular contribution of lung tissue. And so we developed techniques that Ramon described. I won't describe them again. So we could actually do at the cellular level what all of us had done at the organ level. And we thought we were going to find what all the textbooks said, which is that a cell behaves like an elastic balloon filled with honey, a viscous core. And our experiments showed that that was totally untrue, that the rheology of cells is this weak power law behavior that Ramon showed, which was totally inconsistent with the whole literature as people knew it at the time, and totally transformed the field. And that work actually has kind of diverged into a lot of different areas: cancer biology, developmental biology, wound healing, but also in asthma research. Stephen Ahn, who many of you know, showed that airway smooth muscle cells from asthmatics are different than airway smooth muscle cells from normal people. We were able to identify, this long known behavior that a deep inspiration is a potent bronchodilator. We were able to show that that works at the level of the cell. If you stretch an airway smooth muscle cell, it totally relaxes the cell, which then recovers its contractility over a few minutes. That mimics what happens at the whole organ level. So it really opened the window of understanding mechanism at the cellular level, and it has taken off in all kinds of different directions with different labs and different methods. Another important method that would kind of parallel this was traction microscopy. So we can measure the forces that cells generate as well at a single cell, actually subcellular level as well. So at the whole lung level, we have plethysmographs and flow meters, and but there's a total analogy at the cell level, and actually it prepared us well. Our background in pulmonary physiology prepared us well to understand cell mechanics, because the same behavior you see at the whole organ level, you see at the cellular level. And now it's actually gone into actually drug development. We've been able to take some of these methods and turn them into high throughput screening. And actually a very recent development is we've been working with Julian Salway at University of Chicago to try to discover new bronchodilator drugs by scanning large libraries of small molecules. So we actually did that. We discovered a new class of molecules called remodelins, which turned out to be potent bronchodilators. But in addition to that, in animal models, you could show not only are they bronchodilators, but they inhibit fibrosis, and they actually inhibit cancer cell migration. So all this kind of spilled out from that early beginning. And actually, I should add that Daniel Navajas played a crucial role. He teamed up with us early on, and it was great to have him on board. So it continues to be a developing story. I can't even keep up with it all anymore because it's going in so many directions. But just so you all know that it all came out of oscillometry. That's the mother of everything we do now at the cellular level. So I'll shut up. I can talk for hours about this, but I won't. So if anyone has questions, why don't we go ahead and entertain them?
Just a comment. Thanks, Jeff, for mentioning the existence of traction microscopy and many other applications. I had no time in 15 minutes to compress all the literature on cell mechanics, and I focused just on oscillometry, as was the title of the presentation.
Well, I have more of a comment than a question, but my excitement for the topic harkens back to the boyhood of my career. And Jeff, the same time you were mentioning, in the early 90s, when Jeff Maxim and Thomas Schuessler were in the basement of the Meekins with Jason Bates developing what became the FlexiVent animal ventilator. And I was working with the lung explants, trying to make movies of the small airways. Suzanne Kelly, who was the fellow of Peter Macklem, was harpooning frog oocytes. And Suzanne developed a technique where with a pipette, a micro pipette, she could actually measure pressure or volume curves within these frog oocytes. You probably are well aware of that work, but here's an example of cellular lung mechanics in action back from that same era, but not using oscillometry.
Anyone else have a comment or a question?
Yeah, Jeff, maybe I could step in.
Sure.
Thanks, Ramon. That was a nice walk through time in the pathway of connecting oscillometry to cellular oscillometry. Really neat to see that. I want to make a comment on the ubiquitous of the structural damping, like these curves that seem to be recurring in so many systems that we measure. If we oscillate material over a frequency range, we get this relationship between the elastic modulus and the loss modulus, so the real and the imaginary part of the complex modulus, that's quite consistent and shows these frequency dependent power law relationships with frequency. And I guess I'm looking at, maybe to be particular about the measurements you showed, but you showed atomic force microscopy, bead twisting cytometry, but also the optical nano oscillometry, which was very neat. But those are different. The modulus from those was really, really small. Like it was really, really small. And I guess those beads aren't attached to the cytoplasm or the cytoskeleton, sorry. Or there's some difference for the reason why they're so small that I've forgotten. I probably knew at one point, but now I can't recall. But the curves are still there. The curves are still there. The frequency dependent curves are there, even though you're maybe not even tethered to the cytoskeleton. So I just find that remarkable. And I wonder if you can elaborate further. Maybe you know more about those measurements than I do. Ramon, you're muted.
Yeah, sorry. I think it's really remarkable that using, for instance, these three techniques, which are so different in concept and also in the details, they produce the same kind of frequency dependence. So it should be something very fundamental, because the techniques are really very different.
And just to be
Okay,
clear, we find that power law behavior in every adherent cell type. It's a universal behavior of adherent cells, not red blood cells, but adherent cells all have this behavior with different stiffnesses, scaled a little differently, but it's a universal finding.
Yeah, and
Okay, well,
over a very broad range of moduli, like whether you're attached really rigidly to something or whether you are completely disengaged, getting very soft values, it seems to be consistent. So not only over frequency ranges that are very broad, but also over moduli, the things you are attached to or the things you are probing, it's quite impressive. And of course, lung tissue, it seems to be prevalent there as well. So collections of cells and cytoskeleton, extracellular matrix, I should say.
Yeah, well, you mentioned extracellular matrix. It's interesting to mention that I didn't comment that because it was cell oscillometry, but in particular, atomic force microscopy, it's suitable for measuring extracellular matrix of tissues, and in particular, to measure the local stiffness of the tissues. So, because one thing is the macroscopic stiffness of the tissues, and the other one is the local stiffness that the sense cell at the scale of the cell. And I think that it will be interesting in the future. There are already some data to compare in different tissues or in different extracellular matrix from decellularized tissues, to compare the bulk or the macroscopic elastance stiffness and the local stiffness.
okay, great, gentlemen. In the interest of time, we're, I guess, about five minutes over time or behind, which is not so bad, I guess. We've been more loose about the time so far, but I do want to get the last two talks in. So we're going to bring it back up to the whole person level. And I'm going to hand the podium over to Ken Berger, who knows I've been a long fan of the work that he and Benno Oppenheimer have been doing in New York City. So it's a pleasure to welcome you to the seminar. And Ken will talk to us about small airway dysfunction during exercise as a potential mechanism for unexplained exertional dyspnea. And Brian Ross, who's one of our junior attendings at the respiratory division and has taken like a duck to water in terms of oscillation mechanics, is well positioned to be the discussant, as he has a strong background in exercise physiology. So gentlemen, please take over the screen, and we await your talk with interest.
I have to say, after listening to Ramon, I have to start my talk with a quote from the great John Cleese, and now for something completely different. And so thank you so much, Ron, for inviting us. This is going to be totally a departure. And while this title sounds like it's going to be largely related to oscillometry, I do want to ask, is there a role for spirometry in the assessment of small airway function? And we'll come back to that as I go through our presentation and what we did in our study. So, you know we can identify abnormalities in the small airways at rest in symptomatic subjects with normal spirometry. However, the relationship to exertional symptoms remains uncertain. And when I explain this to the fellows, we make an assumption just because we made a respiratory measurement and it comes out abnormal, we don't really know how that relates to the exertional symptom, which happens at a totally different point in time than when we make the measurements. So one conventional approach to evaluating exertional symptoms is really to use cardiopulmonary exercise testing, which is frequently performed. However, the routine protocol is generally limited to respiratory pattern, things like frequency, tidal volume, and minute ventilation, but without any specific assessment of the airway function itself. And so our question is very simple. if we augment the CPET to include evaluation of small airway function, and we can do this, as you'll see, both during and following exercise, can we unmask abnormalities that are not evident with the standard testing and begin to provide mechanisms for unexplained dyspnea in these individuals with normal spirometry? And so we had 3 groups of subjects that we looked at. We had a control group, fairly typically defined as asymptomatic. They all had normal airflow on spirometry. They're lifetime non-smokers and no respiratory medications. We have 2 different patient groups, a clinical referral group that was sent to us with exertional dyspnea, and again, normal airflow on spirometry. This group is a bit of a mixed bag, but they had variable exposure to a variety of inhaled toxins, things like smoking, work-related exposures, environmental exposures, etc. And they were variably put on empiric therapy with things like inhaled corticosteroid and long-acting beta agonists, but they all had persistent dyspnea despite that therapy. Also, because we're in New York and we have a big environmental health clinic, we have a WTC dust exposed cohort. These also have exertional dyspnea with normal airflow on spirometry. This was actually a funded protocol in this group. And so these were all lifetime non-smokers, and all of them were receiving inhaled corticosteroids and long-acting beta agonists, but still had persistent dyspnea. And so we always put oscillometry as the first measurement we do, right? We even have to make the measurement ahead of spirometry, but as you'll see in a moment, we really spent a lot of time looking at spirometric measurements. I will show you a little bit of data for maximal mid-expiratory flow rates. This is not my favorite measurement, but I really want to focus largely on looking for expiratory flow limitation now off of spirometry, not by oscillometry. And we do that based on superimposition of the exhale tidal airflow on the maximal airflow during forced exhalation. So I'll show you an example. Here are 2 individuals, and you see your typical flow volume curve done during a maximal forced exhalation. And here is the tidal volume. And you can see both these individuals, the exhale flow during the tidal breathing superimposes on the maximal airflow. So they are by definition flow limited during tidal breathing. And we can do this assessment as it's shown here at during rest, but we can also do this assessment during exercises. I'll show you in a moment. And our protocol, so we did our resting measurements. We then did a cardiopulmonary exercise test. This is a typical incremental workload test, but we include evaluation of airway function both during and post-exercise. During, we look for dynamic hyperinflation and expiratory flow limitation using tidal flow versus volume curves while they're exercising. And then post-exercise, we look for airway reactivity using both spirometry and oscillometry. And so these are our subjects. This is a bit of a busy slide, but I'll just try to make it simple for a moment. Our individuals are reasonably well matched for the usual age, height, weight, and BMI. They have variable cigarette smoking exposure, none in the control group, variable but very little in our 2 symptomatic groups. We do have some amount of environmental or work-related exposures. There's a few cardiac and musculoskeletal comorbidities, but none of these clinically were felt to be limiting for exercise. And as I mentioned earlier, some variable, forget about variable treatment with inhaled corticosteroids and LABA. So here's our resting lung function looking at the spirometry indices, and you'll notice that there are slightly lower values for FEV1 and vital capacity in our clinical cohort, but the FEV1 over VCs and all these parameters, including the MMES, all of these are within predicted normal range for every one of these individuals. So none of these would meet a definition for airflow obstruction by conventional ATS criteria. When we look at the forced oscillation, you'll notice that we have, again, some variable abnormalities in this group, not terribly different from the control group, perhaps slightly more abnormalities in our World Trade group with respect to the 5-20 and the reactance area. Interestingly, when we look and ask how many of these individuals have expiratory flow limitation by spirometry, it becomes more robust. So this is a simple chart. Each box is an individual, And if they have expiratory flow limitation, I put a color in that box. So in the control group, only 3 individuals showed flow limitation on the spirometry, and we see an enhancement of that both in our clinical referral and our WTC DUS groups. So there is evidence for flow limitation in these individuals that comes out when you couple the, when you add the spirometric assessment. We then go on to say, well, what happens when we exercise these individuals? So how does this then show up in exercise? So each of these individuals, we first look at their dyspnea score on the Borg scale, and you'll see no dyspnea in the control at baseline. They get a little bit of dyspnea on exercise. Our referral group shows an enhancement of that. So they have more dyspnea at peak exercise, but they get to the same point. So their peak VO2 is a very similar percent of predicted, and peak heart rate, very similar percent of predicted. The WTC DUS group starts out with a little dyspnea at rest. They develop more dyspnea on exercise. This turns out not to be significantly more than the control group, but I'll point out that this dyspnea limited them and forced them to stop exercising at a lower workload. So they only got up to close to 80% of predicted for their oxygen consumption and heart rate. So they were symptom limited due to their exertional dyspnea. If we look at the traditional pattern, a traditional approach of looking at respiratory pattern on exercise, and we look at parameters like respiratory rate, tidal volume, minute ventilation, or an assessment of ventilatory reserve by relating the minute ventilation to their MVV, we find no abnormalities in any of these cohorts. So there's no respiratory limitation by the classical means in the clinical referral cohort or in the DUS cohort. I just want to caution you, the fact that the World Trade cohort gets to lower ventilation and lower tidal volume is not an abnormality. It means they have a huge reserve that they've not tapped into. And so they don't have a limitation to exercise by traditional criteria. If we then ask, well, if we look at the tidal versus flow versus volume curves, you can see here in red, these are flow volume curves obtained during exercise in 2 individuals, and they're superimposed on the resting maximal curve shown in blue. If we look on the left, you can see the end expiratory position of the curve is shifted to the left compared to the resting position. So this individual is showing dynamic hyperinflation during exercise. On the right-hand side, you can see when you look at the red flow volume curves during exercise, they again superimpose on the maximal curve. So they have flow limitation during tidal breathing while they're exercising. If we ask how prevalent are these abnormalities, and here's the same chart I showed you a moment ago, you'll notice the control group, we see very little abnormality on the tidal flow volume curves, but we see a tremendous enhancement in the clinical referral and World Trade cohort, largely due to expiratory flow limitation, with a few individuals showing some dynamic hyperinflation. We then went on to look at post-exercise reactivity. And so we did this first with spirometry, which is shown here. And here is shown on the top, the maximum change in FEV1 expressed as a percent change from baseline post-exercise compared to their pre-resting value. And if you cross this sort of 10% threshold, that's the criteria set by current guideline documents to identify airway hyperreactivity. You'll notice a few individuals in both groups where we identify some airway hyperreactivity. Interestingly enough, if you look at the vital capacity in these same individuals, which is shown on the bottom, you see a nearly identical or parallel drop in the vital capacity. So this airway hyperactivity, although we're detecting it by FEV1, is probably consistent with small airway closure as the mechanism for the abnormality. We then also did some oscillometry measurements, and this picture is a little bit more complicated, but what's shown here is the change in resistance here assessed as R5 post-exercise. And when we looked at our data in the control group, we noticed a relation to BMI. So the gray shaded area is the mean value in the confidence interval for what we saw in our control group post-exercise. And you can see that there is, that the change of the reactivity assessed by resistance now increases with increasing BMI. But we can use our control group to identify an upper limit of normal based on a 95% confidence interval. And then we can detect a bunch of individuals in both of our cohorts that show airway hyperreactivity using now resistance by oscillometry post-exercise. I'll tell you that these data here are shown for resistance at 5 Hertz, but if we, this abnormality was coupled by an enhancement in terms of their frequency dependence of resistance and enhanced abnormality on the reactance curve. And so it's probably compatible with the small airway abnormality. And if I then come back to our individuals and ask how many abnormalities do we find post-exercise, again, we see very little hyperactivity by the typical FEV1, but we see tremendous enhancement and small airway hyperreactivity in both of our cohorts. And if you look at this chart, you'll notice that we're able to identify or uncover a mechanism of dyspnea in almost all of our symptomatic individuals that's essentially not present in our control group. And so our conclusions for this is that simply focused airway assessment during a CPET flow at rest and the distributable with small airway hyperactivity following exercise. And these would be little spirometry markers techniques, and these abnormalities were not identified.
Sorry, Ken. Joan, whoever you are, there's an issue with your microphone. Can you please mute it immediately? Hello, Joan?
Yeah, I don't know.
I'm sorry about that, Ken. Okay, go ahead.
All right.
This is a liquid that I buy for her every now and then.
Does anybody in the audience know Joan Kamima and possibly is close to her in the next room, hopefully, and can tell her that her microphone is off, has gone rogue?
Ron, you should be able to mute her.
Specifically, okay, let me try and figure that out, Jeff. Go ahead, Ken. I'll see if I can do this.
I just lost my presentation. Hold on, let me go back to my conclusions. Okay, I did warn you this was going to be something completely different, so let me finish up by just saying we uncover abnormalities on the CPET. This is largely attributable to small airway dysfunction, either during or following exercise, detected largely by a combination of subtle spirometry markers combined with some oscillometry techniques, and they're not identified by standard metrics of respiratory pattern. And these data link the exertional dyspnea now that patients complain of to the resting small airway abnormalities that are now accentuated while or just following exercise. In my cohort, there are numerous factors that could have contributed to this. For example, there's variable smoking history, dust exposure, obesity. These become very important in the clinical arena because they determine and have implications for the optimal therapeutic approach. And so I will stop here and discussion, Ron.
Okay, well, thank you very much, Ken. That was not surprisingly a great talk. I'm going to pass the floor over to Brian, and I'll let you take it away, and I'll go back and see if I can mute the offending microphone.
All right, thanks so much. Yeah, I think we still have some background noise, but thanks again for the excellent talk, and thanks, Ron, for the opportunity to take part in this conference. You know, I think what's really interesting about this, and thanks also for sharing your slides in advance. Really interesting to review. I thought it was quite fascinating that in sort of the universe of your study, you know, all participants of the 3 groups had you know, sort of normal spirometry by FEV1, FVC, and of course the FEV1 to FVC ratio. So I think it's quite fascinating using CPET, which is another interest of mine, to provoke the cardiorespiratory system, and also now using oscillometry to gain a new perspective, in this case, into small airways disease. You know, I guess my one question for you, you showed a lot of oscillometry data in the sort of resistance realm, as well as AX. I was wondering, you know, this was brought up earlier in the conference, about using sort of the difference between inspiratory and mean expiratory total respiratory system reactants, and seeing how that might correlate with some of your not only resting spirometry markers of expiratory flow limitation, but also the dynamic changes that you were seeing during CPET. And that might be because I believe Delac et al. Validated that in COPD. So it'd be quite interesting to see if you could sort of show that in the small airways disease population.
Yeah, that's a great question, Brian. I mean, we have our device is the impulse oscillometer, and it does have outputs for intra-breath data. I frankly have never been able to figure out what the intra-breath values are. If you look at Rafaela's paper, he's very detailed about how he computed the delta R or delta X on his data. I'm not sure I can get a straight answer from the manufacturer as to what they're doing. I don't know if the tracings are, I'd have to go back and see. The current software doesn't allow you to access this as easily as it did in the past. So I don't have it readily available. We have played around with this in the past, but it's an interesting question of how the oscillometry measure of flow limitation relates to the spirometry assessment. I would not be surprised, frankly, if they don't correlate, because you're talking about forced exhalation versus tidal breathing, but I don't know the answer.
I see Dr. Callery, who's very qualified to answer this question as well. So please go ahead.
I'm going to quote about qualified, Brian. It's nice actually to get to one of these meetings at last. Life has been difficult the last few weeks. Surprise, surprise. That was a great talk, Ken. As always, it raises as many questions as it answers, but that's what science is about. Practical bit of information. Impulse oscillometers are not the easiest way to extract data about within breath information. With Ben Arley, we did hack or the impulse oscillometer in Bergen and extracted that data from it and programmed it ourselves so that we could get it out. And I think that's one of the reasons they've made it harder to do that subsequently, because they don't want you to do that. We did get 2 papers out of it, so it was worthwhile. In general, though, because the simple values, you've probably got to take average over several breaths. And if you can get any of that data for within breath, I'd look at the mean value over 10 breaths for inspiration and expiration and do it that way. Taking a value, well, it's very arbitrary what represents anything clinical, but there's stuff in the early papers about that. The 0.28 will be the standard and most robust. The second thing is that back 15 years ago now, with Andrea Aliverti, another ex-Meekins alumnus, we looked at chest wall volume changes during exercise in a variety of settings. And we did find that there were a group of people who didn't hyperinflate their chest walls when they exercised, rather like your WTC people who seem not to hyperinflate but develop flow limitation. I just wondered to some extent whether some of this is an acquired response. Those are people with COPD we were looking at. And it looked as if in the earlier stages of COPD, as the airflow got worse, as you tried to force the system with a CPET test, you wouldn't necessarily adapt in the same way. The striking thing really is why your patients don't hyperinflate. If you've got small airways disease, the logical Dennis O'Donnell's approach would be, why don't they hyperinflate to improve their flows and actually maintain airway patency? And I think if you can get to that, because these are people who aren't behaving like lots of other unexplained dyspnea during exercise, where they have often quite high ventilation, but these people are stopping prematurely during the exercise. And I would like to know why they don't hyperinflate in the face of that mechanical problem. I wonder if you have any ideas.
I don't. I mean, I actually, we expected we would see more hyperinflation than we did, and less flow limitation, right? But, you know, I wonder, when I look at Dennis's data, most of what I've seen published with respect to tidal flow volume curves are in people who already have obstructed spirometry. Yeah. So there's very little, we really could find almost no data for what happens in individuals that we're studying where we think there's a small airway abnormality, but the spirometry, by all the usual criteria, are within predicted normal limits. And so I don't know if this response varies based on the underlying degree of say, large airway abnormality.
Yeah, I suspect it does.
But don't you relate a fair bit of that to BMI? I was particularly struck by your first flow volume loop you were presenting, and these were folks that were clearly overweight, obese, and so that's the kind of pattern you get with them. It's just, but with exercise, but Peter's point is correct, because with exercise, you'd expect them to hyperinflate too.
Yeah, the Danish has shown that the obese people do get dynamic
Yeah.
hyperinflation.
Oh,
They're
yeah.
operating at a lower starting volume.
Yeah, Dempsey showed that years ago
Yeah,
too.
yeah.
But so the question here really is that you've just described a form of exercise-induced bronchospasm, or actually airway closure would be a better term for it. And I think that's pretty novel. I mean, David Kaminsky and I showed with a wedge bronchoscope, you get that with, cold air induction as well. And it's, I don't think it's particularly well recognized. And I'm wondering to what extent that response is related to any of the symptomatology that particularly the World Trade folks have.
It's hard to, we make our inferences guilt by association, right?
Yeah, of course we do.
Yeah, you find an abnormal test, someone's symptomatic, and you go, hey, they must be related. And if I told you it was the shoe size that was abnormally big, you'd say I was crazy. But of course, if there were clown feet, maybe it would produce dyspnea. So we have guilt by association. The unique part, I think, of what we did is we're doing the assessment while we're reproducing the symptom.
Right.
So now we have a direct link between what we're seeing to the symptom onset. And then post-exercise, I didn't get a chance to say this, we didn't do this universally because of this crazy recruitment strategy, but we did give bronchodilators to some of these individuals. And then when we do that, the oscillometric abnormalities return to baseline, and obviously the dyspnea resolves.
Yeah, well, that was going to be my next question, and I'm not at all surprised by that.
So I think there is a link here. Exactly what produces dyspnea? I'm not even sure we know what produces dyspnea in someone with severe COPD, right?
No, we don't really know.
I know people who think they do, but that's not the same thing at all.
It's not at all the same thing. Because we had shown years ago that with montelukast, for instance, that chest tightness could be related to lung volume changes. So in this group, you would expect that symptom to be predominant, but I would bet wheezing has to be a major thing, as you breathe way down there and you're starting to gurgle because of the closure.
Unfortunately, I don't think we had a stethoscope on the chest while we did this. So.
Well, next time.
Next time I do the study, I have to confess, I have to tell you, it may be easier to do these measurements with a wedge bronchoscope in cold air.
Yeah.
Not a simple study.
Just one quick thing. I hope I'm not hogging this, but just one quick thing. Your World Trade Center people, I would guess, must have had CT scans on one or more occasions. And if they've had inspiratory and expiratory CT scans, you might, in that group of people, actually be able to say, yeah, we've got other evidence other than our assumptions based on the physiological inference. These people do have small airways disease and nothing else. So that might also help to factor in and refine the data in that particular subset.
Yeah, Peter, it's a great question. They all have chest radiographs. They get CAT scans if the spirometry is abnormal. So we've published, no, but we did publish a case series of 50 some odd individuals that had restriction on their spirometry with low TLC in the plethysmograph. And we had inspiratory and expiratory CTs and even some lung compliance measurements in addition to oscillometry. And we showed that that was all small airway. And we did see a fair amount of air trapping on expiratory imaging and or bronchial wall thickening on the inspiratory imaging. So we have anatomic evidence of airway in those individuals.
Yeah.
Brian, maybe you could take the question from Larry, and then we should probably hand the podium over to poor Benno, who's probably at this point double booked. Go ahead.
Yeah, Ken, I thought your stuff was fascinating. It was along the lines of the CT scan. Because I wonder if these people have mixed disease, and that they have fibrosis in some areas, emphysema in other areas, and it sort of all goes mixed together when you do oscillometry, because you're measuring it at the mouth. But we certainly see in severe pulmonary fibrosis who come for transplant, who are also smokers, they have emphysema on top and fibrosis in the bottom, and lo and behold, their TLC doesn't look so bad because they've compensated for it. And I just wonder if, especially in the World Trade Center people, that there was mixed disease, and some of the oscillometry might be reflecting that.
There are some individuals that show up with fibrosis, but I have to tell you, Larry, it's the overwhelming minority. I mean, it's maybe 1% or less of our population. All of these individuals have normal chest radiographs straight away. Whenever we've done CAT scans in these, we've never uncovered ILD that we missed on the chest radiograph. So I really don't think, it is surprising because they're exposed to the veritable witch's brew in their exposure, but the overwhelming pattern happens to be a small airway pattern in this cohort. That's what makes them so wonderful for us, right? Because we have a universal group with a small airway injury, and there's very little parenchymal disease. And we exclude all those for that reason.
Okay, well, that was clearly a topic of interest given the discussion. So for those of you who are new to the seminar, you can see that I run the seminar very much with the same philosophy as I run my clinics. All sense of time is suspended in the interest of being thorough and getting to the bottom of what's going on. Benno, I will try and buy my way back into your good graces by offering you a beer at my expense the next time we have a real encounter at a meeting. But in the meantime, it gives me great pleasure to turn the podium over to you for you to give us your talk on the metabolic syndrome that's associated with exaggerated small airway dysfunction and respiratory symptoms in obese subjects. And Andrew Curry, who is someone I've gotten to know through Chung Wei Chow, is the pulmonary fellow at the University of Toronto and is a consummate scholar, I think, and someone to look out for. He wrote that nice paper in Chest looking at the role of or the lack of access to lung function. And he's just finished a manuscript that'll be tucked away because of its length in the European Respiratory Review, but he does a very nice job summarizing the history of spirometry and dispelling some of the myths and taking spirometry back 200 years before Hutchinson, and also addressing some of the issues that Ken had raised in terms of the relationship of race and spirometry. So Benno, it's a great pleasure to have you at the seminar. Please, we are all looking forward to your talk.
Thank you very much, Ron, for that kind introduction. And again, my talk is, as Ken's, maybe a little more clinical in the obese patients. It's titled Metabolic Syndrome is Associated with Exaggerated Small Airway Dysfunction and Respiratory Symptoms in Obese Subjects. And I just want to say that this is a little bit of an excuse, right? Looking at metabolic syndrome at obese subjects has been a topic of mine for some while now, but it's really a topic in disguise. And I'd say the real interest of mine, at least, is evaluating airway function during restoration of operating lung volumes in obese subjects. Essentially because, I've always been confused or at least puzzled or bothered by the fact that obese subjects may have intrinsic lung disease, but they have mass loading with low operating lung volumes, two reasons by which respiratory impedance is altered, and distinguishing both is significantly, challenging. So in this case scenario, I want to introduce some data that we collected in obese subjects and essentially addressing this, right? Obesity is associated with small airway dysfunction that is traditionally attributing to airway compression, reduced operating lung volumes due to mass loading. But there is association of, obesity with intrinsic airway function secondary to this big and pro-inflammatory state, particularly in subjects with metabolic syndrome. The association between the two has not really thoroughly been explored. I think of it as a good opportunity to look at these mechanisms. So in this study, we investigated the relationship between metabolic syndrome and distal airway dysfunction. and respiratory symptoms, particularly. Just like Ken, this is a clinical study. This is, you know, a group of patients that come our way, and we try to take a look at mechanisms of disease. And what we did is observe through restoration of operating lung volumes, through surgically induced weight loss or voluntary inflation to predicted FRC, the effects on removing one of these problems on the parameters that we collect, and see if we can learn a little bit about airway function once one of the problems is partially or fully resolved. So again, why metabolic syndrome again? The adipocytes increase significantly with you know, release of systemic cytokines and you know, significantly enhancing pulmonary inflammation. This is something that Mancuso published over a decade ago. So the intrinsic airway disease is way likely, and that's why the mixed problem is so nice to explore in this group of people. So how do we do this? With a fairly simple maneuver. Essentially, we, you know, take a measurement of oscillometry at baseline FRC and compute the predicted FRC difference, and have the patients inflate their lungs voluntarily and measure the parameters again once patients have reached their predicted FRC. The difference between the inspiratory capacities is obviously the inflated volume, and we assure that we lift the weight off the chest as a means to mitigate the effects of those low lung volumes and their effect on respiratory impedance, potentially uncover intrinsic lung disease and see if that correction brings these parameters back to normal. So how would it look in an obese patient without disease? Well, if we look at resistance at baseline, it's usually fairly elevated, no surprise there. When we restore their lung volumes to about 80 to 100% FRC, resistances come down significantly. And if we now take that measurement and relate it to the lung volume at which it was obtained and compute a conductance, we'll see the conductances remain in a fairly narrow and normal range throughout a fairly broad range of FRC percent predicted. It's a way to relate, you know, those parameters that we see as abnormal and try to understand whether a simple effect such as that of mass loading has an influence. We have a tendency to look at things as percent predicted, don't really pay that much attention of the restoring operating or the operating lung volume at which the measurements are taken, and I think it's an important issue. So what did we do with these patients? So we defined metabolic syndrome using the International Diabetes Foundation criteria, measured spirometry and plethysmography at baseline and post-weight loss, and then assessed some oscillometry parameters sequentially, first baseline pre-op, then following weight loss, whereas the first mass unloading, we gave them bronchodilators to understand their airway reactivity, and then we'll inflate them to predict that FRC. And in all of these instances, try to learn something about airway function until their FRCs are fully restored. We looked at 110 subjects, 73 without metabolic syndrome and 37 with metabolic syndrome. They're pretty similar with respect to age and body mass and prior to weight loss surgery. The metabolic syndrome patients were a little bit older, not too much older, 10 years. And again, I think this is another important point, which is looking at our parameters in the context of spirometry. So in metabolic syndrome negative patients, the lung volumes and spirometry values that we obtain are pretty consistent with what is classically seen in obesity. In the metabolic syndrome, though, forced vital capacity is a little more reduced. Their flow rates are a little more reduced. They have a reduction in TLC and an increase in RV-TLC ratio, suggesting air trapping. And potentially in the context of this normal large airway function, which is well known in obesity, we suggest that these parameters over here suggest greater small airway dysfunction. Obviously, same thing happens in oscillometry, resting R5 minus 20, and reactance parameters are more abnormal in the metabolic syndrome positive patients at baseline. So we could think that these parameters confirm what we're seeing over here and these hints that are observed in spirometry and lung volume, suggesting or maybe even confirming greater small airway dysfunction in these patients, despite the same BMI and body size. So after weight loss, they lose the similar amount of weight. Their BMIs are very similar following surgery, and their FRC is restored to the same degree, not entirely to normal, but pretty much up into the 75 to 80% predicted following weight loss. So there is a significant mass unloading by having them lose weight surgically. So at baseline, following surgery, frequency dependence of resistance that was fairly abnormal pre-op in both groups decreases significantly, albeit a little less, and still with significant abnormalities in the metabolic syndrome positive patients. The same thing happens with reactance area or the reactance spectrum. So these more abnormal residual abnormalities following weight loss in the metabolic syndrome subjects are associated with symptoms. So the lower respiratory symptoms are highly prevalent in both groups at baseline, right? So they all have dyspnea, cough, and wheeze. And post-operatively, there's a near resolution in symptoms in the metabolic syndrome negative patients, but there is still significant degrees of, lower respiratory symptoms in the metabolic positive groups, despite the same degree of weight loss again. And so what we did following weight loss surgery is try to apply these maneuvers. And there was a significant response in all groups following bronchodilator. So here we have the post-op data, where you see the big difference between the post-op R5 minus R20 in the metabolic syndrome negative and metabolic syndrome positive folks. And they significantly bronchodilate after an inhaled BD agent is given. And if we further inflate their lungs and restore them back to their predicted FRC, patients with metabolic syndrome positive, the obese patients with metabolic syndrome that are metabolic syndrome positive still remain abnormal. It doesn't happen necessarily with the reactance area and the reactance spectrum. We're able to correct fully. So these residual abnormalities that are responsive to bronchodilator and correct following an inflation maneuver are significant and very different comparative to the metabolic syndrome negative subject. So in conclusion, we utilize this restoration of operating lung volume by either weight loss or by an inflation maneuver to evaluate airway function in obese patients. We think that the association between the metabolic syndrome and the reduced vital capacity is attributed to the small airway dysfunction beyond the effects of mass loading and obesity per se. We think there is a role for metabolic syndrome in airway disease pathogenesis and obesity, which is further highlighted by this distal airway dysfunction and respiratory symptoms that persist post-weight loss. And these persistent small airway abnormalities are predominantly attributed to a combination of reversible airway reactivity or persistent airway compression. And that's pretty much the clinical data I wanted to throw at you guys and see what you all thought. Thank you very much.
Benno, thank you very much for a simply superb talk. I'm going to turn the floor over to Andrew in Toronto. Please, Andrew Curry, the podium's yours. Lead off the discussion and let's see where this goes.
Thanks a lot, Ron. Everyone can hear me?
Yes,
okay,
we
great.
can hear you well.
Great. And thanks for the great talk, Benno. Made even more impressive by the fact that I believe you just came off of an overnight call shift.
Yeah, I'm a little tired.
Great.
No conflicts.
So, yeah, I mean, again, well caffeinated, yeah. So yeah, no, a fascinating talk, I thought, and a really good example of using oscillometry and dynamic maneuvers to kind of assess airways disease, which obviously is a really cool topic. I think for me personally as a clinician, it was very interesting to think about how this might help us phenotype kind of a difficult to characterize population that we sometimes see, for example, in asthma patients or purported asthma patients who are obese. And I guess maybe can you comment on, whether you think these changes will persist over time, if there's some remodeling going on, and how that might be useful in the future to help us maybe identify a specific phenotype that might be amenable to other, advanced therapies in airways disease, maybe biologics, et cetera.
Thank you, Andrew. Yeah, so obviously, the time it takes for the patients to restore their FRC after weight loss is about 12 months. And obviously metabolic syndrome doesn't just fade away, right? So slowly hypertension, cardiac and pulmonary risk factors start fading as well. So there is a time component involved over here potentially. However, at the same point in time in everybody, we do see these persistent abnormalities. And like you said, there is something else going on in these airways. Restoring the lung volume, the operating lung volume, allows you to see what's happening under the hood in a certain way once the mass loading has gone away. And potentially, yeah, I think that physiological, clinical phenotyping associated, like Ken just showed us a little while ago, the clinical symptoms, the physiological phenotypes that appear, and manipulating them slightly would allow hopefully to uncover some mechanisms that are addressable with newer drugs, or potentially avoiding others such as steroids in people that don't need them, given the fact that obesity and asthma are frequently put together. I don't know if as an intrinsic disease or as an obesity asthma syndrome. That's up for debate and discussion as well, but certainly something that could be looked into and phenotyped a little bit better to hone in on mechanisms and medications or therapeutic interventions a little bit better, for sure.
Great. Any other questions?
Yeah, great talk, Benno. Really fabulous. I missed it. How far after the surgery did you measure? 6 months or is it 12?
No, it's more than 6 months. It takes patients about 8 to 12 months to fully lose that weight. It doesn't happen overnight, for sure. And again, the same thing happens with the metabolic syndrome negative positive patients. They don't all of a sudden miraculously stop having diabetes, hypertension.
Well, that's what we found too. It takes a while, and then they start to come back up. So it's would be interesting to follow them up. But the one thing that really does change rapidly that I still can't get my head around is symptoms. Their symptoms improve right away, and yet the weight loss takes months to really occur.
Yeah.
And then I guess the other point I want to make is that your data is really great because it shows the effect of lung volume and how important it is that we have to start taking that into account in our interpretation of oscillometry. So thank you for that. Great.
Thank you, John. Thank you.
Just to chip in and support, as usual, I normally support everything Charlie says, as he well knows. But
you're going to use that.
Those points. It's a really great study of nature that you've done, a lovely piece of clinical research, which is very informative. I think that there's always a doubting Thomas over here on this side of the pond, and that is a slight concern as to how much is this the metabolic syndrome as opposed to the age of the participants. I just wonder if that lack of reversibility, because you've very elegantly shown you can get quite big improvements in these people with affecting the mass loading, you can get a further improvement with bronchodilators. In the people with metabolic syndrome, you didn't quite see complete correction. But is that because they'd had the problem longer and therefore they had more closed airways? And I wondered if you'd tried a kind of nested matching of people with similar ages anyway, to see whether or not that might influence your final conclusions. But it's lovely work.
Peter, thank you for your comments, and most certainly, and particularly in obesity, there is more to airway dysfunction in obesity than meets the eye. In metabolic syndrome patients, you have to add other things such as pulmonary vascular congestion, leak, alveolar capillary remodeling, and a whole host of things that time certainly affects and potentially have the possibility of turning these things either into remodeled airways that just don't react the same way or take longer to readjust. Like Charlie says, a lot of these patients regain their weight, right? They gain the system by drinking soda and things like that. So they regain their weight frequently. So it's difficult to do that time study. Right? But yes, there is an age difference. Certainly more time to affect the airways, and your point is super well taken. Although they're fairly young, right? So they're between 40 and 50 years old. So it's not that big. They're not that old. But in regards to the potentially still closed airways, yeah, it's possible, but we really made the point to unload those lungs the best way we could, with weight loss and with restoration of lung volume and getting their operating lung volumes back to normal and lifting the weight off of the lungs as best as we can. So as farther than that, I really would not know how to take it. But again, the persistent abnormalities begs the question, as you very well said, is there something else going on? Symptoms improve drastically, but they don't go away in the metabolic syndrome negative patients at all, and they persist, and that is a problem. Particularly, if you just take a look at pool data, obese patients do improve following weight loss in their symptoms, but if you start breaking them down by subgroups, not all obesity patients are alike.
Benno, it's Jeff. Thanks for a terrific talk. Two comments. One is that part of your protocol and the way you framed it was I really liked, and it was kind of reminiscent of Ding, Martin, and Macklem years ago. But that wasn't what we knew things were going to change with lung volume. You're looking for something else. I was just wondering, Stephanie Shore did many studies on obese mice with airway reactivity in response to ozone. I'm wondering to what extent those kinds of studies inform yours or are totally different and are uninformative. Are you familiar with those?
My goodness, you're taking me back a while. And my initial incursions in obesity were in mice models, the OB positive mice particularly. Yeah. And it's a problem because their airways, I would love to bring up pictures. I was forbidden to do so by my colleagues, but because they're so off topic, but the lungs are so drastically affected in OB mice. They're congested. We've given diuretics to OB mice that didn't move in their box, and all of a sudden they're running around like crazy, completely wet, peed the hell out of themselves, and gain activity a lot. So there's so much going on, which is beyond just pure airway disease, mass loading. And it relates to things such as inflammation, alveolar capillary thickening. We can see in microscopy red blood cells moving across the alveolar capillary membrane into their airways. And the mouse model is super complicated. I'm not that sure that I would be able to make that parallel, between mouse and man, if you want to put it somehow. It's difficult. So just looking at the effect of lung volume and restoring it hints to some of these mechanisms that are prevalent in mice and present all at that same time, and dissecting them out might be a little bit difficult, at least for me right now.
Thanks.
Benno, this is David Kaminsky. Thanks so much. That was
Hi, David.
I was fascinated by the fact that your two groups were matched for BMI and weight, roughly speaking, and yet had such different lung mechanics.
Yeah.
And metabolic syndrome obviously is the hypothesized explanation. But within that, did you look at to see what hemoglobin A1C levels were, or glucose levels, or in particular waist circumference? What part of the metabolic syndrome might be affecting that potentially?
It's really interesting. We just published in ERJ a whole, a host of about 400 obese subjects, and the things that really resonate in patients that have airways disease are the presence of hypertension, not surprisingly sleep apnea, and the presence of diabetes for sure. In these patients, we defined metabolic syndrome by those. So the BMI, like I said, not all obesity is created equal. Central circumference is of importance. Metabolic syndrome, cardiac and metabolic biomarkers certainly have a big effect on lung function. And again, I have to use the term terminal lung unit because more than just the airways get affected. And we published the effects of cardiovascular disease on obesity. We do measurements combining diffusion capacity and oscillometry, and it's very interesting how some of these oscillometric and diffusion parameters move in sync, thinking, or at least making this terminal lung unit a composite unit that gets affected in obesity. With the diffusion capacity, you can measure the resistance across the alveolar capillary membrane, the congestion of the pulmonary vasculature. And it's very interesting that in metabolic syndrome patients, congestion is rampant. Membrane diffusion is very, very impaired, and airway disease is very abnormal, or distal airway abnormal. So there's, again, so much more that meets the eye with obesity, and particularly when you start factoring all those things in, certainly.
Leonard, do you want to take the next question?
Thank you. Thank you, Benno, for a great presentation. I thoroughly enjoyed it. I have a sort of question comment here, and maybe more people than you would like to chime in here. I often get questions from people asking me, what would be a good reference for somebody who is really fat and I want to compare it with some normal data. And I've thought about this for quite a while, and I kind of come to my personal conclusion or suggestion that maybe there's nothing particularly normal in terms of being very obese when it comes to lung function, because as you show here, so many different things will affect the lung mechanics. So even if you don't have metabolic disease, and you should maybe be able to claim that, well, this could be a normal person, although BMI is way over 35. So my sort of suggestion would be, could we maybe say that the normal reference value for somebody who has obesity would actually be as if that person had been BMI, say, 30, and that would be sort of the normal, and not try to get normal equations for people who are very, very obese. What are your thoughts?
It's interesting that you mentioned it. I think reference of obesity in population gathered data for standardization and comparison is important. And again, like you just mentioned it, standard equations for skinny adults certainly are biased in obese patients for sure. So Ken, I don't know if you want to comment about the obesity data that you collected and standardized. Certainly, please pitch in. But again, the volume issue and the other aspects of obesity come into play. So it's certainly not a fair assumption to measure those patients with the same ruler.
I think you were touching on something that's I've not heard discussed before, which is how should one incorporate obesity or body weight into a normative equation? And frankly, we don't have a standard approach. So for spirometry, the weight doesn't enter in, and you see reduction in vital capacity, and we say, okay, the vital capacity is a little bit lower than it should have been had this person been thin. And maybe that relates to their symptoms, which could be related to their mass loading. Then we got oscillometry, and then all of a sudden everybody, or lung volumes, and everyone is throwing body weight into the equation. And now you're saying, oh, this is beyond the effects of obesity. The problem is we have to ask ourselves, if we want to know if obesity and mass loading itself is producing a problem, say a symptom, then you have to have the reference as a lean body reference. If you want to ask, is the obesity doing something beyond the mass loading, then you need to somehow figure out how to find healthy obese individuals, which I have to tell you is next to, up to a BMI of about 30, 35 perhaps, but once you get above that, everyone's symptomatic. So
that's really my point too.
So, and I think that there isn't really a consensus as to how to do that. The reference equation I published had BMI as one of the predictors because that's the current vogue, but and we're anxious now to get that programmed into our system so that we can now look at this question specifically, but I don't have that information yet. But I don't know, I think the respiratory field in general should come to a consensus across all normative data as to what approach to take. And you should not have piecemeal where one test does one thing and a different test does something different.
Ron,
well,
mind if I put up 3 slides for Jeff?
I have nowhere to go. So, folks, anyone who wants to stay on, and my hat off to you, Benno, for staying awake through all this after a night on call. The screen is yours again.
So these 3 slides are only for Jeff's enjoyment. So essentially, these are the subjects that we looked at to answer your question. This is an OB mouse compared to a normal mouse. And you can see, obviously, the size difference is huge. And this is what I was talking about in regards to the airway on a macro slide and parenchyma compared to these control mice. These red blood cells and the alveolar capillary macrophages and vascular congestion is present. Their hearts have significant hypertrophy, particularly on the right side with some necrosis here in the RV junction. And if you give them diuretics, their minute ventilation drops drastically with barely any change comparative in the normal wild type controls. So that's just a little tidbit of why things become complicated and airway disease stops being as simple as it relates to BMI. And predictors are difficult. We did publish 3 phenotypes of obesity, ones which are simple obese and have no abnormalities beyond those allowed by mass loading. Those that have abnormalities in oscillometry that are a little beyond, have symptoms, have metabolic syndrome markers, but have normal spirometry, and a group that have overt obstruction. And the group with normal spirometry, metabolic markers, and greater abnormalities on oscillometry at baseline look way more like those that have overt obstructive airway dysfunction comparative to what would be appropriate just for mass loading alone. So if you break those down into these 3 groups, you will find drastic differences in what you would call airway function and the effects the effects on small airway function is discernible by just correcting and getting the mass loading out of the way. But there is more that meets the eye.
Well, looking at those microscopic sections, it makes me wonder when you say the airways haven't reopened as the weight is coming off. Are the airways in fact still there to reopen? Has there been remodeling going on, or has there been actual loss of airway number, such as we see in COPD, for whatever reason, be it chronic inflammation or some unknown mechanism?
Yeah, I mean, that, Ron, is a great question, because the remodeling is certainly something that is possible. it's been described. It is time dependent, and that's why, you know, Peter's question is so relevant. And certainly affects something that Andrew mentioned. Or do we give steroids to these guys, make them more obese, but take care of their symptoms? Or do we, you know, look at these markers, these phenotypes, and say, wait, hold on a second, this is way beyond what is, you know, allowable by the weight and the mass loading per se? Is there anything else? And should Andrew come up with a study that uses physiologic phenotyping to say, you know what, forget the steroids, there's something else going on, and we should do something different. You know, not everything that wheezes is asthma, I was told once, and certainly it might be the case in obese patients.
Great. Are there any other questions before we let Benno go home and get some breakfast and much needed rest?
Ron, I just might add one thing to Benno's talk. Benno probably knows and can. We published some years ago pre and post bariatric surgery, but at 2 different time points, at 5 weeks post bariatric surgery and at 6 months post bariatric surgery. And we measured upright and supine. And what we found, sort of a plug, I guess, for the sensitivity of oscillometry, was that you couldn't see any differences in the spirometry measures or the lung volumes 5 weeks. Well, I think FRC might have been a touch different, just barely. But oscillometry in the supine was quite a bit different, especially both R20 and R5, pointing to something happening maybe centrally as well as perhaps peripherally as well, which leveled off after six months. But you were talking about the peripheral airway function, but there seems to be something early on that happens centrally with bariatric surgery in the very obese.
Okay, great. Well, look, thank you, Benno, and thank you, Andrew. Clearly, the torch has been passed, Andrew, so you've got some grant writing to do. Thank you all for staying on to the end of this marathon of the Oscillometry Summer Seminar, and I look forward to seeing you all again for the last part 4 a week from now. Be well until then. Bye-bye.
Bye, everybody.
Bye, everybody.